All right, guys, moving on to lab 21 and 22. This is where it starts to get a little bit more intense. Can be a little confusing. Of course, rewind the video if you need to, etc., etc. So, page 52 in your lab book. Now we are going to talk about translation and transcription. Okay? So, transcription first. How do we get a piece of DNA turned into a piece of RNA? If you remember in the last video, I told you guys that we started a piece of DNA that looks like this. It's kind of like a wound up ladder if you want to think about it that way. And it unwinds. There's an area that splits open like this where we have bases in the middle. And the cell can come in and use those bases as the template to make a strand of RNA. Now, RNA can be a little confusing because it's not the same as DNA, obviously. There's one big reason for that. Well, two big reasons for that. Reason number one is that RNA is single-stranded. So it only copies one side of the DNA. So I had that picture where it had the two sides that were unwound. Each RNA is one-sided, one side of the DNA, which is really cool because then that means we can hold double the number of genes, right? So it only copies one side at a time. The other reason is that the T base, or thymine, is not in RNA. Instead, we have a base called uracil. So this is what our bases look like. DNA has the original ATGC. RNA is AUGC. So when you guys have a DNA strand and it says use this DNA strand to make a strand of RNA, you need to remember there are no T's, okay? So if you guys get this piece of DNA up here, this is DNA at the top. We'll label D for DNA, R for RNA. Be very, very careful about when you're doing questions on your test, perhaps. Be careful. Is the question asking you to use this DNA to make another strand of DNA, or is it asking you to take the DNA and make a piece of RNA? Because that base is different, because the RNA has the uracil instead of the thymine, the answers are not going to be the same. So you got to watch it. So this is the piece of RNA that's made. Over here, if you look, right up here, we have a U instead of a T. This particular stretch that I drew just doesn't happen to have any other U's, but if there were more A's up here, there'd be more U's down here. It's important to note, if there's a T in the DNA, there's still an A in the RNA. It's only the T that has changed. Okay? All right, let's see. Next thing. There was a question on the lab. Why does DNA have to be transcribed into RNA? Why can't we just go straight from DNA to protein? There are a couple of answers for this, even though this question was technically extra credit. There are a couple of answers. One is that the DNA is inside the nucleus. The DNA is a really big molecule, and there are pores in the nucleus to let things in and out, but the pores are really small. So DNA can't actually get out into the cytoplasm where our ribosomes are, and ribosomes are too big to get in. So it's one of those things where we have to kind of find some sort of segue in the middle. DNA can't come out, ribosomes can't go in, so we got to do something. And RNA is a small enough molecule that it can leave the nucleus. So then why do we have a nucleus? Like, why wouldn't we just get rid of the nucleus? Well, the DNA needs to be protected. It has to be protected, and it needs to be easily found by the cell when it's time to make more DNA, make proteins, make RNA, etc., etc. So it needs to be easily accessible and not floating around in a place where it could get damaged. So then, let's see. Oh, this confused a lot of people, which is fine, but we're going to go through it. So, transcription. The DNA unwinds, gets copied into a piece of RNA. The RNA leaves the nucleus, goes out into the cytoplasm, and finds a ribosome. And this is where translation comes in. Okay, write everything backwards again. Well, forwards. Okay, that's better. So, translation. We are in the cytoplasm. We've got our RNA. Now, if you guys look, it says mRNA, and all that means is mem stands for messenger. When an RNA molecule is getting ready to leave the nucleus to go out into the cytoplasm, it goes through some processing. A couple of things are added to it to make sure that the cell doesn't go, hey, that needs to be cut up and destroyed. It makes it so that the body's like, nope, I need to use this for a while. Leave it alone for now. So we go out and we find a ribosome. Now, a ribosome is made up of two subunits, a big subunit and a little subunit, and the big subunit sits on top of the other one. And we have a couple things that happen here. The ribosome will start moving down. You can see it's kind of like a sandwich. It's kind of like an RNA sandwich. It starts to move down the RNA, and it reads those bases. The bases that we got from the DNA as a template, it starts to read these. It reads them in sets of 3. And that set of 3 is called a codon. So what happens is our ribosome scoots along, and there's one specific codon that triggers that ribosome to start making a protein. In the RNA, that codon is UAC. These over here, these squigglies that I drew, are what are called tRNA molecules. And these guys are interesting and very, very important. So this ribosome is scooting along. It sees a codon that says, hey, it's time to start making a protein. What happens is that when the ribosome finds that codon that says, hey, start making a protein, there's a tRNA molecule that matches up with that codon. So here, our UAC matches with this AUG. So this guy is going to float up and take this circle right here and put it into the ribosome. The ribosome, once it starts reading more codons and grabbing more tRNA molecules, is going to start taking these guys and linking them together. So over time, there are many different codons, and each codon codes for certain things. There are multiple codons that can code one thing. Some codons have, one pattern for one codon. It just depends on what it is. And all of these things are amino acids. And amino acids are what proteins are made out of. So our ribosome is scooting along, reading all of our sets of 3, our codons. The matching tRNA, whatever it happens to be, goes to the ribosome to drop off its amino acid in the right order. The RNA sticks all the amino acids together in the correct order, and that is what makes our protein. It's complicated, I know, but that's what makes our protein. So if this guy was going along, and there are 20 different amino acids that we use. Some of them we make ourselves, some of them we have to get from our food. But either way, there are 20 of these guys, and in many, many different combinations, that is how we make all the proteins in our body. Pretty cool. So, now, we have gone from our DNA to our RNA, and we've gone from our RNA to our protein. So, how do we know which amino acid is which thing? Now, there's a picture that I know was included on your guys' quiz, but I like this one better. I know everything is backwards, I'm sorry. So this picture is actually in your lab book. It's the back of page 55. And I like to read the wheel. I personally feel like it's a little bit easier to read. If you guys come out here, so you can kind of see the letters. I know that the actual words are backwards, but bear with me here. So if you're looking, let's say there's a list of amino acids. You remember I said there were 20 of them. Their names are all around here. All around here. So if you're trying to find a specific one, you look around the wheel, try to find it, and you're like, okay, well, this one is called threonine. And so it's like, okay, well, this is called threonine. Which RNA codon codes for threonine? How do I get threonine? The big is in the middle is the first letter of your 3 letters. Next one is your second letter. Next one is your third letter. So threonine right here is A, C, and it is actually coded by UCA and G. So there are 4 different codons or 4 different patterns of letters that can give us threonine. So another example. There's Here we go. Here's a good one. Up top, up here. This one's called methionine. This is the codon that every single protein always starts with. So if we read this, how do we get methionine? A-U-G. Hopefully that makes more sense now. So if I were to say I give you a strand of DNA, and I say, take this strand of DNA and write what the RNA letters are supposed to be. And you do it. Okay. Then we take those RNA bases, divide those up into our little 3 sets, our codons. These 3 are together, these 3 are together, these 3 are together. And I tell you, take those and figure out which amino acid corresponds to them. That would be a good example for something that may be on your test. So you should be able to go that direction, but also reversed. If somebody gives you, a list 5 amino acids, you should be able to go back and figure out the RNA molecule, and even further back and figure out the DNA molecule. It takes some time. It's true, it takes some time, but it's doable. Okay? So don't get discouraged. One step at a time is the key here. And now there are a couple of other examples that I'm going to go through. Just little things. I won't need the board for this one. A couple of things that was in your guys' lab, and y'all did pretty well with it, but I want to go over it anyway. So on the back of page 52, there are a couple of questions about bases and how they're kind of ordered. So there are a few different kinds of mutations that we can get when we're, translating DNA to RNA, or transcribing DNA to RNA, or translating DNA to proteins. So what can happen is if we have our string of bases and we're just moving right along and everything's great, we have our string of bases and something happens. Either there's a mutation, something gets moved where it shouldn't be moved, something like that. There are a couple of different things that can happen. One of the things that can happen is that one base can get substituted for another. So if there's supposed to be a C, maybe it gets changed to an A, something like that. Sometimes that doesn't actually affect the amino acid sequence. If you guys look at the circle chart that I was just showing you, there are several different codons that code for the same amino acid, and that's part of the reason why. Even when there is an error, you can sometimes still get what you're supposed to get and get a functional protein out of it, which is pretty cool. It's kind of a way for us to safeguard ourselves. However, sometimes that doesn't work out. Sometimes a base changes in a spot that just doesn't work out, and it changes that codon, meaning that we get a different amino acid in that spot. So let's say instead of a threonine, we get a valine, or instead of a threonine, we get another methionine, something like that. That's going to mess up the protein when it's finished. If you guys remember, one of the big things about proteins is their 3D shape. Their 3D shape is very, very important. And if even one amino acid in a 3D protein shape is wrong, it can completely change the shape of the protein. A good example of that is the disease known as sickle cell anemia, if you guys have heard of it. It is a disease where the hemoglobin in an individual's red blood cells is mutated, and hemoglobin is the part of the red blood cell that picks up oxygen from your lungs and moves it around the rest of your body. So it grabs onto the oxygen and, transports it around. Their hemoglobin has a mutation in it. It has one single amino acid different than what it's supposed to be, and it completely changes the protein. It makes it so that it's very difficult for their bodies to transport oxygen. It actually changes the 3D shape of the red blood cells themselves, and that's why it's called sickle cell anemia, because they kind of look like sickles. They're like moon-shaped, and actually because of that shape, it also does damage to the liver. So it's not great. That's just an example of something that can happen from even one single mutation. And when one nucleotide base changes like that, we call that a point mutation because it's just one spot. There are a couple of other ones. There are what are called insertions and deletions. So an insertion is just, this base got stuck into this sequence where it wasn't supposed to be. So if we have a string of, let's say, 9 bases, which would give us 3 codons, right? We have a string of 9 bases, and a 10th base just gets thrown there in the middle. It's going to push all the bases after it back 1, and it's going to change all of the codons after that mutation, because now everything's out of order. It's like taking the numbers 1 through 9 and then sticking a 12 in the middle. It messes it up. Same idea with the deletion. If you delete one, it pushes everything up a letter, and then everything's out of order and all of the codons are different. So sometimes this produces a long protein that just isn't what our body wanted. Sometimes it accidentally produces what's called a stop codon, which is literally, this protein is finished, stop making it. So we have the methionine, which is called a start codon, which is what all our proteins start with, and then we have stop codons. And sometimes when we have an insertion or deletion, or even a point mutation, a codon that was supposed to be for something else gets turned into a stop. So we end up having a protein that's too short and can't do its job, which is bad. I think that's pretty much it. Yeah. So remember, DNA gets transcribed into RNA, gets translated into a protein. There are U's in RNA, not T's. There are tRNAs that bring our amino acids to our ribosomes. Yes, it's kind of complicated, but I have every faith in you guys that you guys can get through this stuff. This is the last video for today for me. I've done all the rest of them that I needed to do so far. I will probably do more videos for your next assignment, Wednesday's lab, which is evolution. So that'll be fun. I am going to give you guys some extra time to do the genetics lab and the evolution lab, because I know you guys have a test this week for Dr. Merchant, I do believe. And then what I'm going to have us do is, and unfortunately there's no really other way to get around it, we've brainstormed, and it's just going to have to be, you guys are going to have to film yourselves presenting your cool creature and send me a link on YouTube. Now, I know that sucks, and I'm sorry that that has to be like that, but that's the best way to do it. Unless you really want to do it live for me over Zoom or something like that, upload it to YouTube. What I am going to ask you guys to do is I'm going to have it due on the 10th, which is a week from this Friday. And the reason I want to push it back, because technically it's supposed to be due on Wednesday, but we're pushing it back just because of all of this stuff. You guys, a lot of you guys, I think, have had to move back home, move out of the dorms when you weren't originally planning it. There's just a lot of stuff going on. So next Friday, April 10th, is your due date for the assignment. I'll put a thing on Blackboard where you can just send me the link to your YouTube video. If you make a YouTube video, it doesn't have to be public. You can make it private so that the only people that can see it are the people you send the link to. And then after you get your grade, if you want to delete the video, you can. Totally up to you. It's fine with me. And then I will upload your rubric. So I will take notes and grade everything as I'm going through your project, and I will give you guys feedback, and we'll make sure that you guys get your points and everything like that. I'm trying my best to keep up with grading on Blackboard. It's a little bit faster, I think, than hand grading. So it's going pretty well. If you guys have any questions about anything, any of the labs, the quiz, anything like that, please reach out. I check my email pretty frequently, or at least I try to. If you really can't get a hold of me, you can text me. My number is in the syllabus. Yeah, so I will probably have another video or two for you guys on Wednesday for the evolution lab. In the meantime, please work on your stuff. Please stay safe. Stay home. Don't go out if you can help it. I know the urge to do so is strong, especially since it's been a couple of weeks, but it's very, very important that we keep ourselves and everybody else safe. Okay? I hope you guys are doing well, and I hope you are staying home, because I certainly am. But yes, be safe. Wash your hands. Don't cough on anybody, and don't let them cough on you. Okay? I'll see you guys on Wednesday.